My name is Elijah Torbenson. I'm a third year medical student at Creighton University School of Medicine. And before medical school, I was a student at the University of Minnesota, working as a bioinformatician under the mentorship of my principal investigator, Xiao Dong. And I joined his lab with an interest in senescence. And so my initial project was working on developing an analysis pipeline that would be able to better identify senescent cells in single cell RNA sequencing datasets. However, as I began working on developing this pipeline, I was quickly both amazed and a little bit overwhelmed at the sheer number of analysis tools available, with oftentimes no clear way to evaluate which ones would be best to use in my pipeline. Not only that, I would get different final results based upon the different tools that I would use. And so my research interest actually pivoted to the development and comparison of these tools themselves, such as where Steve comes from. And so the ultimate goal was to develop tools and pipelines that would be able to detect senescent cells. But this is kind of um, along this process, developing and comparing these tools is kind of where my research interest shifted, and that's where Steve came to be. I would put it kind of like this. When doing a single cell RNA sequencing experiment, even if you perform the techniques perfectly, the end result is still a computerized data file, like a sequencing results that you need to process. And this process has several steps. And at each step, there's a myriad of computational tools that you could choose from that are available online. And so rather than being a tool you use at a specific step itself, Steve is more so a way to help researchers evaluate whether the tools that they used in the pipeline are good tools holistically. And so it's able to evaluate the pipeline as a whole, as opposed to doing a specific function at a certain step, which is what most tools you find online will do. So, for instance, data normalization, you'll find tools that will help you with that. Feature selection, clustering, all these different steps, so we can find different tools online to help you with. You'd put those all together to form your entire pipeline. And what Steve does is it helps you look at the pipeline holistically and helps you evaluate whether it's a good fit for your research goal. Steve, indeed, the ultimate goal was to refine these pipelines so that they can better be used in research itself. And so, in our instance, we're talking about here helping us better detect senescent cells. Um, During my time there, I spent most of it working on Steve. And so, as I've kind of graduated now into now in medical school, I'm not directly involved in senescence research at this moment in time. Um, but as my lab continues to develop pipelines that can better detect senescent cells, I believe that Steve will be used to better help in that process. And so Steve itself has been a work in progress. It's still been, even since I've left, we've been kind of working on it during the past couple of years to refine it. And it's getting close to being pretty much done. And it's, we submitted it to our journal. But that's all to say that it's just gotten to this point, and so it's not seen too much use yet. But that's what that we anticipate is that it will be used in helping develop pipelines. So senescent cells are very interesting to study, but sometimes they're difficult because their relative sparsity in tissue can make it hard to detect them. In addition, they can be very heterogeneous. And so that's, for instance, why single cell RNA sequencing is one of the tools that are very useful in studying them. The issue is once you do single cell RNA sequencing, you have your data, you're still not sure which cells are which. And so generally, if you have a pretty well-known cell type, if you're just annotating, for instance, B cells or adipose adipocytes, that's pretty easy to annotate. But it gets to the point when you're looking at extremely rare cell populations in that tissue that it can be difficult to identify them. And so what we're hoping is that um, pipelines in the future will, with more certainty, be able to detect senescent cells in these RNA sequencing experiments, and kind of organically, not simply just doing a like a bulk RNA sequencing where tissues were treated with some damaging agent and a lot of the cells are assumed to be senescent. This is more so looking organically in tissue, looking for those rare senescent cells. And once these pipelines are able to do that, I think we'll be able to better characterize senescent cells and understand their expression. But that all rests on the premise that you can, or that you're able to identify these kind of rare cells in these tissue. And so, um, I'll start talking about more, a lot of these pipelines, they need to be more further optimized to successfully detect rare cell populations because senescent cells would fall under that category. So Steve has four modules, and when researchers are using this, they're able to use all the modules, and they're also able to choose which ones they want to use that best answers their question. And so, basically, giving us just a little overview here on what each module does. The first module, called Subsampling Evaluation, um, its purpose is to assess pipeline stability across sample sizes. And so that will make more sense as I go more into it. But the second module is Novel Cell Evaluation, and it assesses the pipeline's ability to detect new cell types. Or I can also say rare subpopulations of cells. And so this would be particularly relevant with senescent cells. Um, the third module is Annotation Benchmarking, and so it's able to assess pipeline accuracy overall. And then the fourth module is more of an outlier. It doesn't really fit with the first three, whereas the first three are used to evaluate the pipeline. The fourth module is a function that Steve does itself. And so, although Steve is used to evaluate the entire pipeline, part of its intrinsic judging capabilities comes from its ability to annotate cells itself. And so users are actually able to use Steve if they don't have a cell annotation tool that they like. They can just use Steve directly to annotate their cells as well. And so that's an optional capability. However, the main purpose of Steve is to evaluate the pipeline. And so the fourth one is definitely the most optional out of all of them. So moving into the first module, this is kind of like a theoretical pipeline a researcher might use to process their data. This is just a visual graphic to help understand this concept. So you start with your data from some sequencing, and then you do some sort of processing, and that data is processed. You process it again to get your final results. And so for our cases, to keep things simple, let's say you start with your sequencing data from your single cell experiment. You perform data normalization, which is an extremely important step in processing the data. And then the next thing you might do is you might annotate the cells. So I'm skipping some steps here, but this is just for example. And so you annotate the cells, and your final result is a data set which has the cells annotated. And this here is just a visual representation in a reduced dimensional space of what the data set might look like. And so you have each individual dot here is a cell in the data set, and as the researcher using Steve, you would have gotten to this process already. And so you would have been able to annotate each of these cells. And so the cells in yellow are these classical memory cells. Cells in blue are these double negative cells. And now the question you might use Steve for now is how stable are these annotations? Specifically looking at this step right here, the cell annotation step. However, by extension, you're also asking how stable is the data normalization step? Because the premise of Steve is that all upstream processing steps will have an effect on downstream results. And so, for instance, even if you change nothing about the cell annotation step, and you chose a different tool for data normalization, you might get different results, even though you keep this step the same, because this upstream step does have an effect on lower downstream steps. And so Steve is focused specifically on this part of the process, the cell annotation part, but by extension, it really is a reflection upon all upstream steps because upstream steps will change the final results in the cell annotation step. And so this is basically how this module one works. For input, you take your data set, which has already been annotated, and the researcher has already annotated it using their pipeline that they want to evaluate. So they've chosen their data normalization tool they want to use, and they've also chosen the cell annotation tool that they want to use. And so they input that into Steve. And as output, Steve puts sensitivity and specificity metrics. And so we'll get into what these actually mean. And so how this works is the data set that Steve has taken for input, Steve randomly samples 10% of that data set and sets those cells away. And so this is represented here by 10% of this data set being put away here. And the remaining 90% are right here. And now what happens is the remaining 90% of the data set is used to annotate the cell types of the 10% through the use of Steve's novel and intrinsic reference mapping function. And so this is what I was talking about earlier. Steve has intrinsic reference mapping function that can be used to annotate cells, but this is, as opposed to that being the purpose of Steve, the purpose of Steve is to evaluate this entire pipeline. And so once again, in this process, these remaining 90% of the data set is used as a reference to annotate this 10%. And now these new annotations of the 10% that came from using this 90% as reference are compared with the original annotations. Because once again, the researcher has already annotated all of these cells using their own pipeline. So what Steve does is it splits it into two, uses this 90% here to annotate the 10%, and then those annotations are compared with the original annotations. And that's where you get your sensitivity and specificity metrics. And this is repeated across different splits. And so right here we have a split of 10% versus 90%, but it gets repeated so that it's 50% and 50%, all the way to 90% and 10%. And so your final result is these sensitivity and specificity metrics. And what you can tell here in this specific example, the relative stability of the sensitivity and specificity measurements here would suggest robustness in the pipeline, even in conditions of limited reference data. So for an example, an annotated single cell RNA sequencing reference data set of adipose tissue will have few senescent cells in total because they're relatively sparse. However, we would expect that we could still use that data set, given that those senescent cells were annotated, as a reference to identify senescent cells in other data sets of adipose tissue, despite those senescent cells being relatively few in number, because of the relative stability of these metrics over these different splits. And so that is where this module can be useful. Another point where it can be useful is it can also be used to evaluate the quality of a reference data set. And so, as I've kind of alluded to before, a lot of the ways you annotate cells is you have a reference and then you map those reference labels onto the new data set. A lot of that process is dependent upon the quality of that reference data set. And so this module can also be used to evaluate the quality of that data set. And so whether you're using the Tabula sapiens data set or a data set that was annotated through the help of experimental means, for example, it's fluorescence associated cell sorting, this can be used to help evaluate whether that reference data set is a good quality data set. Now, moving on to the second module, this is called novel cell evaluation. And so the point of this one, as I talked about previously, is to help tell whether your pipeline is able to detect rare cell populations. Because sometimes the issue is when you're trying to identify a rare population of cells, instead of the pipeline recognizing that that's a new cell or a rare population of cells, sometimes it overfits the annotation process, and those rare cells just get misclassified as some of the more abundant um, cells. For instance, that would be the case of a cell just being, instead of being correctly identified as a senescent cell, it gets identified as a healthy adipocyte because adipocytes in that tissue would be much more abundant. And so this pipeline evaluates the ability this module evaluates the ability of pipelines to detect these rare cells, and by extension, for our cases, senescent cells. So it's a similar process to the first one, except here it's 50% of the data set is sampled, and then the remaining 50% is set aside. And this is where now it also departs from the previous module. The set of cells that are being used as a reference to annotate the other set, they have all these different cell types. And so, for instance, here we have the classical memory cells, we have naive cells, we also have transitional cells. In this module, the transitional cells in this reference is intentionally removed, such that the corresponding cells, and so here I have, this is 50-50, this is 90-10, but it should be 50-50, such that the corresponding cells, the corresponding transitional cells in this part should be annotated as unknown or novel because the reference that is being used to annotate this part no longer has those cell types in its reference. And so the question you're looking at here is, will this pipeline correctly identify those cells as being new cells or unknown cells because those cells are not found in the reference, or will it misclassify them as other cells that are present in this reference? And so this is this here is an example of what you might have. And so um, here are the different cells. And lower metrics indicate an inability of the pipeline to identify novel cell types. And so these rare cell types would likely be rather classified as an already existing, more abundant cell type found in the reference. The third module of Steve is annotation benchmarking. And so as I talked about previously, there are reference data sets online that are in some ways considered benchmark or gold standard because there's a high confidence in their annotations. And so, for instance, data sets validated through experimental methods, for instance, fluorescence-associated cell sorting, where cells were physically removed from each other based on cell markers, then sequenced, you have a high confidence that those cells are from that cell type. And so data sets composed through those means, you're pretty confident that they're good quality. And then other examples of this would be these large consortiums of data, for instance, the Tabula sapiens data set, that was annotated through many experts collaborating with each other. And so there's also relatively high confidence in those annotations. And so what researchers can do is they can take those data sets online and use those data sets as a reference to annotate their data sets. However, in the case of Steve, the way that Steve utilizes these data sets is they what Steve does is it takes the user's pipeline and using the user's pipeline, it annotates the, I guess I put it this way, the researcher will use their pipeline to annotate that benchmark data set. And then what Steve does is it takes those labels that was created using the pipeline we're evaluating, and it compares those labels with the original annotations. And once again, because the original annotations of those benchmark data sets are, we have high confidence in them being accurate, we can thus have a idea of the accuracy of the pipeline in annotating that data set. And so this one here uses some of these available data sets. And Steve, as a package, does come with these data sets available. And so that should be relatively easy to do in the experiment. Finally, the fourth module is, I didn't have it here because, once again, it's more of an outlier. Steve can be used as an annotation tool itself. And so if, for instance, the users want to use Steve, they're totally able to do that to annotate their cells, given that they also have a reference. And the questions we're researching are so, the field of senescence is so impactful, and even doing a really good job with the single cell experiment, it's so important to be thoughtful in the data processing steps. Like I've said previously, you could do the single cell experiment perfectly, and we're still not at the final product because you have to go through all these analysis steps. And so what I would say is, the results that researchers find from this research are important, and they've also put a lot of work into these experiments. And, you know, it's important to continue to have that quality in the data analysis phase. And so tries to help researchers with that because they put so much work into designing these experiments. Um, you know, why not, you know, why not have help moving forward in the data analysis phase two to assure quality throughout and then confidence in the results. And so um, hopefully it will help researchers um, both researchers in the analysis, but also researchers who design and do these single cell experiments because um, the results that you get are heavily dependent upon the tools that you use. And so I really hope that Steve will help people in this process.